My name is Arian Avalos. I'm the research geneticist here at the Baton Rouge Lab. Today I'm going to be talking a little bit, sort of the broad landscape of how we're using, how we're considering breeding and then genetics within breeding for honeybees, how the current state of things as well as moving forward. So, one sec. There you go. All right. So breeding is a relatively old practice, and it stems naturally. It's as old as animal husbandry and farming, and it stems naturally from these approaches. Because if you can think about it, the moment you start keeping bees, or the moment you start planting crops and things like that, you naturally, if you're interested about the organism and you get to know the organism, then it flows naturally that you are going to start then asking yourself, how can I improve aspects of what I get from this organism, or maybe improve the organism such as better health or longevity, etc. So we call this incidental selection, sort of like the beginning of sort of what a breeding program may be, and this is both in history as well as presently how many breeding programs starts. And I would say most of you are likely familiar with the process that's associated with this type of selection, because it's pretty intuitive. So you have a population of bees, for example, and then within this population, you have a particular subset of bees that you're really interested on. This is sort of like what's the black bee that's circled there. And you're saying, okay, well, I really like the particular characteristic or characteristics of this bee. We call those traits or phenotypes. And so I take these bees and I isolate them and I then propagate them. That just means I make more of them. So that's what the bigger circle shows. And then the next generation hopefully has more representation of that type of bee in it. And then you sort of rinse and repeat on a yearly basis, right? We're talking about in history as well as sort of most breeding programs. This starts just with a beekeeper's or a bee breeder's hunch, let's say. Right, as in like I feel these bees are productive or they're good for what I want them to be, so I'm gonna go ahead and maintain these bees, right? And this is really good. This is a really good sort of general outline and a robust one, because honestly, it's even the general outline that we follow in more advanced sort of breeding practices. But it has some limitations. So this is a cyclical process. It's gradual. It's linear. That means that you have to measure the trait that you're interested in on a yearly basis. It's time consuming, so it will take the year to often measure many of the traits. So like honey productions, for example, we're lucky here in Louisiana, as you've heard, and that we get multiple honey flows, for example. So that's something that is different from other parts of the country, where you sometimes only have one time in the year where you can measure honey production, really. And then it's time consuming in terms of man hours, and it's costly in terms of funds as well, right? So it's a year-to-year process, and you can only do usually one selection bout per year, which then determines how fast you can improve that bee, right, or that population of bees. Well, this is where it logically falls into sort of like what the reviewers in this article like to call breeding 2.0, because scientists are not immune to buzzwords. And this is sort of like general localized improvements on sort of specific aspects of this major outline. So as you saw there, the two major components of incidental selection and then breeding in general really is picking your bees and then making more of your bees, right? Well, in breeding 2.0, you're improving this because you're incorporating statistical and experimental approaches, but then you're also improving sort of the scalability and logistics. So you're tackling both of these simultaneously. How can I improve the way in which I pick my bees, and then how can I improve the ways in which I make more bees, right? So then that, like in one side of it, right, how can I improve the way in which I pick my bees? That's where sort of we start with adding experimental approaches, as well as basic things like good record keeping, right? And I cannot stress enough how good record keeping informs a lot of the breeding decisions that we have here at the lab in general. And so to give you an example of this, in the bottom graph there, you have an older study done in the lab where we compare Italian stocks to Russian stock in terms of overall growth rate across multiple years. Well, because we've been able to keep consistent and good records from year to year to year, we can definitely monitor this trait and then tell you, is it stable, is it improving, is it increasing, things like that, right? And incidental breeding, so breeding the 1.0, I would say, because it's more on an individual basis, sometimes the records are not good, sometimes you're picking something that's slightly different, and so it models a bit on what type of bee it is that you're getting generation after generation of bee, right? On the upper graph, you have another type of question that then we can ask if we add experimental sort of approaches to this, and this is like, how stable is my trait? Here, this was another study done with the VSH bees, where the lab had an ongoing population. We gave it to honeybee queen producers, and then told them, well, give us what you sell to the beekeepers after you've done whatever it is that your operations does with these bees. And so when we got them back, we saw that the trait actually was highly variable, which is very stark contrast to the population we have here at the lab, where it was still quite robust, right? And so that helps us because it tells us, well, somehow the trait is not stable once it reaches the hands of honeybee queen producers. And so we need to figure out if that's a part of the genetics of the trait or if that's a part of the operational approach. And then how can we then work with the producers to get maybe better management practices or more trait aware management practices, right? So, but all of this is really just focusing on how do we pick our bees, right? And then the other side of the coin, which is how to make more of our bees. Well, this you've heard a bit on previously with Kate and Garrett, in that it's centered more around, you know, management and sort of efficient practices, right? So things like having a mating yard or a mating area in order for you to be able to control reproduction better, so that helps you maintain your trait a little bit better. Having a queen bank, for example, so that you can tier up the way in which you assess your queens on a seasonal basis. So approaches like this help you help you go from a sort of, you know, small scale breeding operation to a larger scale breeding operation. And that has many benefits. It's not only the benefit of being able to produce more bees of a particular type, but it's also allows you a larger population where you can take a look at and say, well, are there any new traits that I might be interested in, right, that I maybe have missed because until now I've been keeping, let's say, 100 colonies. Now I have 1,000. I definitely can probably see more nuances and more traits that are working better, or new traits that I wasn't even considering before, right? So that scalability allows us to feed back into the system and sort of make a better breeding program. To highlight something, this is the status of bee breeding, definitely in the lab right now, and I would argue probably in the nation as it stands, right? And that's fine, it's fair. This is where we're at in terms of like the situation, but where we want to get to is sort of the next step, and we call this breeding 3.0, again, buzzwords. And all breeding 3.0 does is it incorporates genetic information. So this is more of what I've been working here at the lab to move more towards, essentially, because I do think that this brings, I mean, it's not only my personal opinion, it has brought immeasurable benefits to other practices. For example, having this incorporation of genetic information in your decision making for breeding has improved corn yields by 4-fold over the course of, you know, a handful of decades, essentially. And I think there's a lot of benefits we can reap from this in the bee breeding world. So to put it in perspective, what is then breeding with genetic information added to it? Well, like this seems a little bit daunting, but I'll walk you through it. The figure seems a bit hairy there, but all this is saying, well, it's taking that basic cycle of picking your bees, making more of your bees, doing this year after year, and actually splitting it up some by taking advantage of sort of the genetic information of the bees, right, which acts as a sort of barcode. For the bees of interest. So we split that up and we say, well, what we're going to have is a training cycle. And the training cycle is going to be just us figuring out what part of the genetics of a particular bee are tied with what we're interested in that bee. So once we make that tie, once we know which markers associate with a particular characteristic or particular set of characteristics, let's say, then we can say, okay, we have our predictive model. So now now we can, we don't have to measure the trait. We know that reliably we can predict that the bee is going to have these sets of characteristics about 75, 90, 80% of the time by just looking at the genetics of the bee. And so what that allows us to do is then go into a selection cycle that only uses genetic information. And so you then have a population of bees, and instead of having to phenotype every single bee on a yearly basis, we don't do that. We just take the bees, sequence them, and then from there come up to a final outcome that is probably, or it should be at least, very similar to if we had done that traditional sort of linear cycle. All right, so what are the benefits that bee breeders and beekeepers could reap from this sort of approach? And just genetic information in general? Well, as I mentioned before, the standard breeding cycle of selection and expansion, essentially, it's a lengthy cycle, right? There's shortcuts that you can take sometimes, but generally speaking, in bees, we're talking about a year's worth of work, and it's every year, right? So for a small scale operation, this might not be a problem, right? You can just keep making your breeding decisions as they go. If you're just really, looking for your own type of bee for your own practices, that's not a problem. But you can see how this could become a potential problem if what you're doing is you're trying to provide a service of queens to a broader, like more expansive, let's say nationwide population, right? So time is a limitation here for sure. The how to pick your bees, like, right, so like that first phase, that also has a time limitation, right? Many of the traits that we're interested in, definitely here at the lab, but broadly speaking, take weeks to months to phenotype. Much of the time is wait time. Other parts of that time is just, there's only certain times in the year where we can measure these things, right? So for example, for aurora resistance, whether you're looking at aurora resistance as a blunt measure, or whether you're looking at aurora resistance as a rate of growth, so in the first scenario, you have to wait until late in the season because those levels of mites are more predictive of survival for the colony after the winter ends, right? So that's a year you had to wait in order to figure out whether your colony is resistant or not, right? So for varroa growth rate, right, we are measuring it twice during the year, once in spring, once in the fall, but those two measures actually are one measure because it's a rate, right, is how much it increased or decreased, etc. So again, you're spending an entire year measuring twice a year, but you're spending an entire year to get your one result, right? And so this is one of the other limitations, is that this particular selection cycle takes time, right? And then the other thing is that once you're done with this entire process, the story continues. So as an individual running an operation, you're going to have to keep doing this year after year after year in order to make sure that your trait is stable in your population, right? Which again, for a small scale beekeeper, not that big of a problem. You can work it within your, within your yearly work cycle. But for a large scale beekeeper, this becomes prohibitive, immensely prohibitive, with the more colonies you incorporate into your operation, right? So these are perspectives that we have to consider here at the Baton Rouge lab. So then what are the benefits we can reap from sort of the integration of genetic information? Well, like in the first phase, you're not really gaining a lot because you're still having to phenotype your trait, and you have to link that trait to the genetics, right? So the genetics is relatively quick. You can sequence a bee relatively fast, but the phenotyping is what takes the time. Where you save time is that in this model, right, in this approach, you're only really having to do this once every couple years, right? So you go from having to do this sort of like phenotyping every single year to just doing it once every 3-5 years, let's say, in your population, depending on how stable your trait is, depending on how accurate your predictive model is. And so you already save, 2 years worth of time and resources, right, by just being able to make this association, right? And then the other benefit is that, well, okay, once you have that association, right, you spent a year, you've built that predictive model based on the genetic information, then you only use the genetic information, and you can use that genetic information actually agnostic of the trait, right? So you don't have to wait to measure varroa at the end of the year, let's say, or varroa levels at the end of the year. You can just in spring sequence, let's say, drones or daughters or workers of particular queens that you might be interested in, And then so in order to identify whether that queen might be a particularly good candidate for moving on in a breeding phase, and then you can just make your breeding decision in the spring right there, and then move on and have your next generation. And the benefit of that then is, I mean, and that can take anywhere from days to weeks or months, depending on how fast the sequencing service is. But for the most part, the key point here is that you could do this anytime in the season, right? So as a breeder, this gives you, again, a leg up. You're doing it earlier in the season. That means you get your queens in, you make your breeding decisions rather quickly. You don't have to like wait on the previous year's measurements. You kind of already have your panel going. And then the other thing is that you possibly could gain even more, because if you can do it early in the season, that means that you can feasibly then take those daughters, the result, and then do it again a little bit later in the spring season, in the summer season. Then you can have 2 generations worth of selection within a single year, whereas before you were restricted to really 1 generation in a single year, right? So these are, these are incredible benefits to large-scale operations and potentially to smaller-scale ones or intermediate ones as well. And they, all towards the goal of breeding for a better bee, whatever that better bee means for you, right? And in the case of the lab, like we mentioned, like you heard from Garrett, and again you heard from Kate, better bee includes bees that are both bees that are selected for individual traits such as varroa resistance or varroa sensitive hygiene, for example, with the pole line and helo ideas, but also means breeding for better bees, in a general sense. So Russian bees, for example, is more of a panel of traits that they're interested in, a pedigree, let's say. And so that's their idea of a better bee, and so they breed for that. And I will say we do use genetic information for the Russian honeybee program, but right now it's not quite at this level. So we're kind of working to make it a bit better in that regard. And then we hope that with the Helo program, we can maybe see about how we can incorporate this to then expedite that process as well. And so these are sort of the types of ideas and sort of the goal to get to in the lab. And you heard a little bit from Mike in terms of like, what have we done so far to kind of get there? And so this year we published a paper. You're seeing the first figure of it. Again, it looks a little bit hairy, but all it is panels A, B, and C just show the relationship, the relationship based only on genetic differences between commercial stocks and then research lines. And in the research lines, you have pole line, you have Helo, you have Russian, and Minnesota hygienic as well, even though it's not technically anymore a research line, but more of a broad scale available line. But then you have a couple other major sort of Italian beekeeping or bee stocks essentially. And so A, B, and C describe how these individual stocks are related to each other, sort of how they differ and how they're similar in terms of genetics. And then panel D shows you the broad genetic groups and potentially that there is actually structure within these groups that we have to be mindful of when we're trying to quantify or generate that model based on genetic associations, right? So this is our first toes dip into this realm right now, but I will tell you there's actually several continuing studies which include getting more of this type of information so that we have a better reference panel that we can feed back on and potentially, even help further with. So like, if you have a trait of interest and you want to see if there's other bees across the states that might have the trait, that might have a similar trait or might look like the bee that you have, that you might want to incorporate because you're worried about inbreeding or something along those lines, well, then you can, talk to us about, if your bee looks like something in the reference panel, and then we can maybe potentially talk about how you can maybe get some of that material and things like that. The other things that we've been working with, so this is, again, scary plot, but it's really not. All this is an assembly report, and this is basically a very genetics-oriented figure, but all it's showing is a new reference assembly for the honeybee genome based on one of the commercial stocks. We're actually building several of these assemblies. They're not as high quality as the current reference assembly, but we're planning on using them to then create something that we like to call a pan-genome, which is sort of like a joint reference genome that we can then, will be improved from the current one, as well as include more information inherently about different types of populations of bees. And so this is, I'm excited about this. This is actually in a collaboration with Dr. Brian Scheffler. and Justin Vaughn's the part of ARS. Brian is in Snowville, Mississippi, and so we're steadily working towards getting more of this information so we can establish both a better reference and a better reference genome and a better reference panel, and then from there on it's just a matter of building better predictive models, which is kind of what I'll also be working on independently. So with that, I might have rushed things a bit, but if there's any questions, feel free to write them up. Or if you have any individual questions to me, by all means, feel free to email me at arian.avalos@usda.gov.